Hello everyone, this is my project on using computer vision to quantify behavioural correlates of sublethal pesticide toxicity in bees. So pesticides, while they're obviously revolutionising pest management for farmers, it's also led to a huge overuse and misuse on farms, with techniques such as seed coatings or spraying pesticides that have been major contributors to certain bee population declines in the world. And these pesticides are particularly bad for bee health as they mainly target the nicotinic acetylcholine receptor. This is found in the insect's central nervous system that binds to acetylcholine and facilitates signal transmission in the insect nervous system. With most of the popular pesticides acting as competitive modulators to this binding site of this receptor. However, the pesticides don't impact all insects the same. That's because of a couple of things. Oh, wait. Oh, yeah, and non-lethal concentrations can cause these behavioral changes in bees when they don't kill the bee. Um, Pesticides don't impact all insects the same in this case, though, because there's a couple of things. The first one being that there's a massive diversity of this NACHR receptor across insect species. um, with each insect species typically having their own unique subunit in this receptor that serves functions unique to that species. And ultimately, that can impact how effectively the pesticides can bind to this receptor. Secondly, different pesticides have different affinities to this receptor. So, for example, sulfoxamines and neonicotinoids appear to bind similarly to this receptor, but it's been shown that sulfoxamines have a lower affinity for binding compared to most neonicotinoids, and that typically makes their effects less toxic on insects. And also, some species of insect may have certain metabolic enzymes, and pesticides might follow different metabolic pathways that allow them to detoxify certain pesticides. A good example of this is the honeybee that expresses genes for the cytochrome P450 enzyme that can detoxify flupyrafurone, which is a type of pesticide. And finally, different pesticides at different concentrations can impact species differently in terms of sublethal concentration effects. But at the moment, there isn't really any overall consensus on how to quantify sublethal effects. There's been obviously a lot of studies done on sublethal effects, but there hasn't been many to do with across different toxins or different pesticides or across different species. And this is because how the main way to study these so far have been using markers, which are very time consuming, and they're also quite challenging to implement without impacting behavior of the insect, and therefore they aren't very scalable. And as I said, it makes it hard to study these differences in pesticides and differences across species. But computer vision might be a really good solution for this, because it doesn't have the challenges that are involved with using markers, which makes it a very good contender to study variation in sublethal toxicity behavior studies. So Kieran has sort of looked into this before, and he very nicely did the data collection that I'm using for my project. So thank you, Kieran. I'm guessing most of you already could know what Kieran's done, but just for context of the project, or if anybody doesn't know, I will go over it very briefly and oversimplifying what he did really. But he used these three different pesticides at these different concentrations of the LC50. And what the LC50 means is basically if I get a sample of bees and I give them 100% of the LC50, you would expect 50% of the bees tested to die. And so what Kieran did in this data collection is that he put these bees in this perspex box with this feeding hole where the feeding tube would be. That's how the pesticides and the sucrose to keep the bee alive would be administered. And this box would go into this ethoscope here, where there's a camera attached to a Raspberry Pi, which is a mini computer essentially, and that records all the data from the camera. And so Kieran put these bees in a 24 acclimation period before he actually did any testing. And this is just to make sure that the surprise of being put in a confined space didn't impact the results. Um, stop the bee from going bonkers just because it was in a confined space and not because of the pesticide. And so when Kieran exposed these bees to pesticides a day later, he monitored these in 5-minute segments, recorded them with the camera, with 20-minute breaks, and that totaled for about 24 hours per bee tested. And so with the data Kieran collected, my goal was to advance the work that he did by using machine learning software called Deep Lab Cut, which is just a labeling software like Animal TA, but Animal TA is a more manual way of doing it. And the goal is basically to process all of the data that Kieran had collected in a feasible amount of time. And then once I have got the data from Deep Lab Cut, my second goal then was to get any behavioral markers via processing this data that might be associated with different sublethal amounts of pesticides, with the potential of these being concentration dependent. And with all the 3 pesticides that Kieran's tested here, they all work fairly similarly in that they bind as agonists to the nicotinic acetylcholine receptor, and basically essentially overload the nervous system. So I think we expect overall to see some increase in activity here. Granted, there might be some differences because sulfoxaflor and fluopyrifurone are typically weaker in interaction with the receptor compared to thiomethoxam. And radfurone has different metabolic pathways to the other two, and that might impact as well how the bee behaves when treated with them. So, Deep Lab Cut is the software I'm using. As I said, I think a lot of you are already familiar with this, but for context of the project, I'll just go into it briefly. So, Deep Lab Cut uses deep neural networks for markerless pose estimation and posing estimation, in the form of point coordinate data of animals from videos, and this involves labeling a given amount of frames from given videos, training a model on these already labeled points, and then the model can be approved upon if the tracking of the model isn't good enough, or the model can be used to analyze a group of videos with the trained data set you give it. And once fully trained, Deep Lab Cut in theory can analyze any video you give it. And some studies on rodents indicate that training on a few annotated frames around 200 can be good enough for good accuracy, and it's very quick to analyze a big amount of videos, which is what I needed, because there's about 6,000 videos. So this is why I felt like Deep Lab Cut was a suitable choice for this. And this is just a general overview of my project. I'm going to first talk about the pre-processing of Kieran's data and just the Deep Lab Cut stuff. So, as I said, I had to downsample Kieran's data. Originally, it was a 1080p at 60 FPS, the videos he recorded. And the reason why it had to be downsampled that much, as you can see what it says there, is because initially when I trained this model, it was taking gauges. And to analyze all the videos with the original model, it was going to take about 3 to 4 months, which obviously for a master's project isn't ideal. So that's the reason why that was downsampled. And also I moved it over from my computer onto Arc, which is the uni's supercomputer. And for my final model, I ended up on 3 points per body part except the wings. The wings were very hard to track, so I just left them. The reason why there's 3 points and not 1 each is because it kind of acts as a scaffold just in case 1 point isn't necessarily tracking very well. So let's say we have a leg and 2 of the points nearer the body tracking quite well, but the end point isn't tracking as well, which is kind of what I've seen. What the model can do in theory is kind of use those 2 points as reference if it's confused on the last point, and kind of just stick it in distance with the previous data, and it seemed to be quite effective. I had to train it on more frames than the studies show, and Again, that was done on rodents. This is a bee, so maybe it's a bit more complex in its movements and how you track it. I'm not too sure. But overall, I got a very confident points from the model. Um, this video that I'm showing you here is all the points are visible if the confidence is greater than 0.6, which is fairly confident, and most of the points are there most of the time. So a very accurate model, and it's very confident on the head, abdomen, and thorax. I would say about most of it I've seen the most confident 0.8, 0.9, so it's very, very accurate. And there's just an example of what Deep LabCut gives you when you analyze the videos. So it gives you every point. So head B is the start of the head, and it gives you the X and Y coordinates for every frame. And it also gives you the likelihood for each point of plot. So how confident is it in plotting that point? And so with that data, I could use this into a program called MoSeek. I don't think a lot of you know what MoSeek is, so I will go into it in a lot of detail. Um, MoSeek is a form of unsupervised behavioral analysis using machine learning to identify any typical behaviors during any analyzed videos using the key point data. In this case, provided by Deep LabCut, but in theory, any form of key point data processing would work. It's been mainly used in rodent studies up to this point, but the people who made MoSeek said that they don't see why you couldn't use it in non-rodents. And the little diagram below just kind of gives you an overview on how MoSeek works. So first you have your Deep LabCut coordinate data by frame by frame, and then you put it into something called noise calibration, which is where you fine tune the noise calibration parameters to smooth over any jittery or high frequency points using a generative modeling approach, and that basically differentiates any actual behavior signals to noise. And you might be wondering why there's a massive cross there, and that's just because the way to optimize this, which is kind of getting example frames of very jittery points and then recorrecting them, doesn't seem to be working in Jupyter Notebook for whatever reason, or it's not compatible with Arc. So I couldn't tune the model's parameters to get the best noise calibration. So that's at default, so it's not perfect, but it will work fairly well enough. And then it's passed into a PCA analysis, which basically just simplifies how many data points there are, and it reduces the data to its most informative components, and therefore removing any redundancy that isn't necessary. And then it has to be taken from this continuous form of data in the form of point coordinates to something that is basically discrete in terms of behavioral units or behaviors. And to do this, we have to pass it into something that's called an autoregressive hidden Markov model. And I'm just going to give some context of what an autoregressive hidden Markov model is. And basically, an AR-HMM segments behavior into short recurring patterns called syllables, aka different behaviors. And basically, the process of this the goal is to define the boundaries and shows the rules governing how these syllables connect over time, aka transitional states, and that's known as behavioral grammar. So a normal hidden Markov model kind of assumes that there are these hidden states, in this case it's behavior, and these change over time based on transition probabilities, and that each state has a observable output based on a probability distribution. So to put that more simply as an example, let's say a bee is resting, for example, and the model would assume that it has a 30% chance of staying still, I don't know, a 50% chance of walking, and a 20% chance of flying, based on its current state, which is resting. But the issue with this is that a normal hidden Markov model doesn't have any sort of memory or doesn't use any past data, and so it treats observations independent as each other. So this is why an autoregressive form of this is important, because the way the probability of behavior is happening is now incorporating the bee behavior before it. So for example, if a bee is slowly moving forward, It's more likely to keep walking based on previous data rather than just randomly flying, for example. And it's more realistic to use this because bead movements typically have smooth transitions, and incorporating past tracking history just overall makes movement patterns more accurate and easy to segment overall. So, enough of that jargon. Um, the deep lab cut output data that I used with MoSeek, um, my goal was to capture any behaviors or behavioral syllables that occurred with a minimum frequency of zero point 0.5 across all the data. This was just the default that MoSeek gives you. And the reason why I left it at that because I thought that um, obviously that's enough to give us indication of the common behaviors, but also we would want any rarish behaviors to be picked out, but not so rare that it's not really relevant. And so from doing that, there were 83 syllables in total, but only 19 of them that met this requirement. And these are just the 19 here. We've got examples as slightly curved walking, normal walking, climbing, flying, turning very sharply, and being still, and these obscured areas, which I'll go into in a minute. And this is just one of the examples from across the videos of one of these syllables. This involves a lot of walking and turning, which in the video you can see is what it's doing. They're all doing something very similar. This is a very accurate syllable. And this is an example of where MoSeek and noisy data is kind of an issue, where MoSeek isn't able to fully separate behaviors apart. So this is describing three different behaviors here. Some of them are being still, some of them are fidgeting, like here, some of them are just moving very slowly. And I think this is ultimately because the noisy data, when the bee is being still, might be associated with a little bit of movement still. And so this is why. And then this is the obscured arrows I was on about. And the reason why this happens is a few things. So there's a lot of areas in the box where the bee can hide under. So this feeding tube, this microphone, I think it is, or there's tape here. The bee can hide under that, and it does in some of the videos. And Deep Lab can't handle that very well, and so the data is very noisy. And also, sometimes it's too dark for Deep Lab to pick up on, and so you get these obscured errors, which happen more often than I would like it to, but it is what it is. And this is just an example of what you can do with the syllable data. So MoSeek will assign a frame with a syllable number, and that can basically be used to plot frequencies of each syllable happening for all different treatments. So this is control in all the TMX concentrations. This isn't a final graph. It's not a great graph. It's what MoSeek gives you. I'm going to do my own, but it's just an example. For example, for TMX 0.5, there's significant difference in, I think that's curved straight walk. So it's a walk, but there's a little bit of turning involved. And you can see that TMX tends to do that more than the rest of the treatments. And that's just some of the examples that you can pick out with this. And then this is just a dendrogram that MoSeek gives you, where it might be evident that MoSeek might not be separating these behaviors fully, and some of them are quite similar to each other. So, for example, syllable 15 and 16 are very similar, 3 and 5, 4 and 7, 6 and 8. And this gives me the potential to maybe merge these syllables together. But MoSeek has no option to do this, so that's going to be a manual process, and that will be something that will require a lot of trial and error. So that isn't finished. The results aren't exactly there yet, but it's like the last thing I have to do. But the rest of this I've got results for. So again, with this coordinate output, I can use it to calculate velocity frame by frame. And how I do this is that obviously from the data, Deep Lab gives you a 2D output. So it gives you an X and Y coordinate, and you have to calculate velocity separately for each axis. And to do this is very easy. You basically just take the coordinate value of the next frame, and you take it away from the coordinate value of your current frame and divide it by 2. And and then you basically have to do an order of magnitude calculation with the coordinate of x and coordinate of y, and that gives you total velocity, technically. And then with this, I can do one or two things. The first thing being that I can plot average velocity per treatment over 24 hours and kind of compare them. And this is for sulfoxaflor at different concentrations. Um, and just to describe the graph, because it can be a little bit confusing, the x-axis segment number is essentially time. I forgot to change this, I'm sorry. This is essentially over the 24 hours that Kim recorded each bee. So, for example, segment number 1 is basically the average across each 5-minute video for that one treatment. And for a given treatment, the velocity is averaged across all the different bees that were tested with that treatment. Um, and then obviously we've got speed. The area around the lines are basically standard error, averaged across all the bees that were treated with the same treatment, again, at that segment number. And just for all these graphs, I'd just like to note that early days of this testing, where we've got segment number 0 to 10, 0 to 15, might not be indicative of anything or relevant, because this is where Kieran gave the treatments to the bees, and as a result, it might take some time for the pesticides to really take any effect on the bees, and so we might not actually see any kind of behavior showing from that. But after that point, we start to see some effects here. So, for example, in sulfoxaflor 0.5, we see a period of increased speed where all the other treatments have crashed. And then further on, we see that sulfoxaflor 0.5 seems to crash harder than the control near the end. And we also see at lower concentrations that it keeps a constant slowness to it, whereas the control kind of boosts near the end in speed. And for thiomethoxam, this is one that was very obvious what it was doing. And we can see that TMX 0.5 has a huge increase in speed. But like the one before it, there's a massive crash at the end where it kind of keeps up with the speed of the control. And we can see that the lower concentrations of TMX don't really appear to do much different from the control. And then with a flupyrifurone, this is a very messy graph. If anybody's got any suggestions on how to plot this better, I'd take any advice on. I'd be more than grateful. And I think overall, you could say that higher concentrations of flu seems to decrease average speed or not change it much. The only reason why I say take that with a pinch of salt is because this flu one here, Kieran only did tested with 2 bees. The rest of these points are anywhere from about 15 to 20, I think. So this one isn't really fortified by a good sample size, so I'd take these results with a grain of salt. But the lower concentrations of flu, we see similar patterns to what the other two were doing at high concentration, where we don't see any crashing at this midpoint. But then near the end, we do see some sort of crashing. And this is just kind of a table that summarizes what I've said in probably a nicer way than I have. But I won't read it again, just for time. And then the final thing I can do is calculate max velocity every 10 seconds and use something called the Cosinella framework. And the Cosinella framework was shown in 2023, I think, by these people at Imperial. And what it basically is just a low computational power way of analyzing behavior in fruit flies with a high throughput. And basically, they use low resolution tracking with low res cameras, and they use real time approximate location tracking, and they real time calculate the max velocity every 10 seconds. So, unlike Deep Lab Cut, this is a very quick way of tracking what the bee's doing. And it uses ethoscopes, which is where the ethoscope name from Kieran's data collection came from. And it works in exactly the same way in that it records the videos of these flies doing whatever. And as I said, it was used on fruit flies. And basically what they were doing was trying to compare behaviors across different pharmacobehaviors over time. And you can see from here is that it's 42% accurate. And what this matrix is basically telling you is that they were feeding the data they got from this max velocity every 10 seconds, and they were kind of training a predictive model, which I'll explain in a minute. And they were basically testing on some data. Can you predict what treatment we had given this bee? And it seemed to predict very well what treatment they gave the fly, 42% accuracy. In comparison to more computationally intensive things, um, it was way more accurate, like 16%. So I followed this framework because I thought it'd be really good if I could prove that it was accurate enough for Rachel or Kieran to do something like this without having to process the data afterwards. And so the framework basically goes as you calculate the max velocity. In my case, using the deep lab cut data, because we didn't use real-time tracking, I had to filter in case of any noise, and I just did this with a median filter, and I would then interpolate any values. And then I had to do what was called a time series feature extraction, called catch-22, across every time series, a time series being 5 minutes each that Kieran recorded. And basically what a time series feature extraction is that it's describing it condenses a long time series into 22 features, and a time series feature basically describes how that time series behaves. So for example, one of the features is autoregression, and autoregression is basically how predictable your data is. And let's say the velocity is always the same in a time series, autoregression is going to be very high because you can predict that usually it will be the same. And so for each catch-22 feature extraction I did, I labeled that with the appropriate treatment concentration label that Kieran tested that particular bee on. And then I would split this into something called test and training sets. And I would use the training sets to train what's called an SVM linear classifier. And this is the predictor that I mentioned before. And basically it just finds the best line to kind of separate the data into different groups based on their labels. And then it kind of uses the line to predict the group of test data that I give it in the hope that it will match to the actual labels that I gave it before. And the reason why it's good to see if it predicts this well is because if it can predict across different concentrations and different treatments, there must be something about the pesticides that affect max velocity in these bees differently, and therefore there must be some indication of different behavior across these treatments. But unfortunately, we saw that it wasn't as accurate as this Cosinella study. So from this matrix, this y-axis here is basically what the actual label of the test set that I gave the SVM linear model. This is what it should have predicted. And then this x-axis is what the model predicted. And you can and this line, this diagonal line is basically correct predictions. And then anything outside of this is not very accurate. These are wrong predictions. And it's not very accurate overall, but there are some treatments that are more accurate than others, but not accurate as a whole. So we've got sulfoxaflor 0.1, we've got TMX 0.5 that seem to be a little bit more accurate. And the reasons why this could be the case is because, as I said, with the MoSeq stuff, we've got very noisy, at times occluded and dark data, which wasn't accounted for here. We've also got a lower sample size. What I mean by this is that even though technically we given the SVM linear model the similar sample size to the Cosinella study, the time series length is much shorter. So in the Cosinella study, I believe they either used 3-hour or 12-hour time series, and we only gave it 5 for the sake of getting enough samples to give the model. And also the last thing that could be the problem is just that bees and flies are different. And you might be like, well, yeah, obviously, no kidding. But I mean that in terms of maybe giving toxic treatments to bees at sublethal concentrations might affect bees differently to flies in terms of maybe it doesn't affect their velocity as much, which is still a good finding to find, I suppose. And that's all I've done. Thank you for listening, and thank you to Rachel and Kieran, who have helped me so much throughout all this.